Hi. In the previous session, we have discussed industrial strains. We have seen the ideal characters of a industrial strain, and we know the methods to occur industrial strain. We can either get it from a culture collection or isolate from the nature. The methods used to isolate an industrially important microorganism from natural resources is called screening. As we discussed earlier, screening has two different stages: primary screening and secondary screening. In this unit, we are going to talk about primary and secondary screening. In the primary screening, we are detecting and isolating the microorganism which has potentially interesting industrial applications. In secondary screening, what we are doing, we are evaluating the real industrial potential of the microorganism. Or other words, in the primary screening, we are looking into the microorganism, like whether the microorganism producing an industrially important compound or it is having a capability for biotransformation. We are looking only at the ability of the microorganism, whether it is doing it or not. But in secondary screening, we are doing a detailed analysis. We have already know the ideal characters of industrial strain. We are comparing the newly isolated microorganism with the ideal characters, whether it possesses all the ideal characters to be used in the industry. So if we jump into the primary screening can be done in two different methods. Either we can do it like direct selection, and second is indirect selection. Direct selection is based on the ability of the microorganism. Suppose if I am going to isolate a microorganism producing antibiotic, what I do, I take the soil, take the samples. It can be a soil sample, it can be a water sample, or it can be air sample. Whatever is the sample, I isolate the microorganism from the sample and look into each of the microorganism, and or I devise a testing technique to see that any of this microorganism is producing an antibiotic, and all the microorganism which is producing antibiotic will be selected for secondary screening. That's the way the direct screening work. So whatever may be the samples, it will have a billions of microorganisms. So I am applying this test to each and every members of this microorganism to see that any of these members, irrespective of the taxa, irrespective of the species, is producing an antibiotic. That's the criteria. And whatever organism producing antibiotic will be selected for secondary screening. In case of indirect selection is basically is based on the taxonomic classifications. As we already know that the group Streptomyces is producing many different antibiotics. So this many of these species of Streptomyces is known to produce antibiotic. So if I am going for an indirect selection, what I do is like I take a sample, I isolate all the Streptomyces from the sample. Then I test the ability of the isolated Streptomyces to produce antibiotic. I am not testing the ability of all the microorganism. I isolated only Streptomyces from the sample. Then I am assessing the ability of the isolates for antibiotic production. That type of selection is called indirect selection. Indirect selection is like a reservation. We are not testing all the microorganism in the group. We are testing only a particular taxa. If you are looking for antibiotic, we can look into Streptomyces because that group is known to produce many antibiotics. If we are looking for a xenobiotic degrading microorganism, it's better to look into Pseudomonas. Pseudomonas is known to degrade many different xenobiotic compounds. So based on the taxa, the isolation happens, and we are testing the ability of these isolated members for the industrial application. So that's the difference between direct selection and indirect selection. And this primary screening can be done in two different methods. First one is detection isolation, which is mainly done in solid cultures like agar cultures, which is used for the isolation of microorganism producing antibiotics, metabolites, or organic acids. And indirect selection is mainly done using enrichment culture. That's a liquid culture. There are exceptions are there, many exceptions are there, but generally the detection and isolation, we use the solid cultures, agar cultures, and it is most probably that will be a direct selection method. And enrichment cultures are used to be indirect selection methods in liquid culture. We can see the details of each and every aspects. So first one is primary screening for antibiotic producing microorganism. I hope most of you have heard about crowded plate technique. When we are plating the petri plates in the laboratory using spread plate or swab, we know that the ideally a spread plate should contain 30 to 300 microorganisms or 300 colonies. Then only we will get a isolated colonies. The colonies will be equally spaced, or we can see the distinct colonies, the number of colonies should be 30 to 300 in number. But if you have more than 300 colonies, the plate will be little bit crowded. There is a technique called the crowded plate technique, in which the number of colonies per plate will be between 300 to 400. That's why it's called a crowded plate technique, which is used to identify the microorganism which is producing antibiotic. In this method, the colonies are so close together. If any of these colonies is producing an antibiotic, what happens is that the neighboring colonies will be inhibited. So we can see a zone of inhibition. That's actually a method for primary screening for antibiotic producing microorganism. Here's the steps what we follow for the primary screening of antibiotic producers. First, we will take the samples. Sample can be a soil sample, water sample, or air sample, and then diluted. Serial dilution can be done to get 300 to 400 colonies per plate, so that it will be crowded. Then once done the dilution, we'll go for a spread plate on an agar plate. After spreading, we will incubate in the incubator, and after incubation, we will look for incubation zone or zone of incubation. If any of this microorganism is producing an antibiotic, the neighboring colonies will not be able to grow. So there will be zone of incubation. In the next slide, you can see that, yeah, here we have the center, there is a microorganism. Because of this microorganism, around this microorganism, there is no growth. This is called a zone of incubation. What does it mean? The microorganism is producing something to incubate the growth of neighboring microorganism. Uh, Sometimes in our laboratory also we have seen this type of zone of inhibitions, but it need not be always antibiotics. Sometimes this organism producing some acids, so the pH will be reduced so that no microorganism is able to grow around. In some other cases, the organism will be like they're very hungry, they will absorb all the nutrients from the surroundings, so the no other microorganism will be able to grow. But in some cases, it is producing an antibiotic. So we can isolate the microorganism and characterize it to see that whether the microorganism is producing an antibiotic or not. So this is the primary screening method for antibiotic producing microorganism. This method is called crowded plate technique. Secondly, the method we can this method is used to the isolation of metabolites producing microorganism. The extracellular metabolites like vitamins and amino acids have very high pharmacological value, and it's also used in the food industry. So there are microorganisms which is able to produce this vitamins and amino acids extracellularly. This is a method used to identify those microorganisms. So I'll just go through the steps. Taking the samples from soil, water, or air. Then we dilute it to get isolated colonies. In the crowded plate technique, the number of colonies per plate we intended to be between 300 to 400. But in this case, the number of colonies should be between 300 to 30 to 300, so that we can see the isolated colonies. Then after dilution, we are spreading it on an agar plate. The peculiarity of this agar plate is that the agar plate do not have the metabolite what we are going to we are interested in. If you want to isolate a microorganism which produce vitamin B12, the plate should not have vitamin B12. It should be a minimal medium without vitamin B12. So only the organism which produce vitamin B12 will be able to grow in that medium. So the agar medium in which we are plating the microorganism should be a minimal medium, and it will lack the metabolite what we are interested in. So after the spreading, we are incubating it with a test organism. So we are inoculating the minimal media plate with the sample and the test organism. The peculiarity of the test organism is that it's an auxotroph. It grows only in the presence of the specific metabolites. If you are looking into, if you are if you are trying to isolate a microorganism which produce vitamin B12, what we do, we take a plate without vitamin B12, and our test organism will only grow in the presence of vitamin B12. So by default, the test organism will not grow in our medium because it needs vitamin B12. So what happened is like during the incubation, if the any of the microorganism present in the sample is producing the vitamin B12 and it excreted out to the medium, the test organism present around will be able to grow. That's the only case the test organism can grow in the medium. So if any of the microorganism present in the sample is producing vitamin B12 and secreting out, then what happened? The test organism can use this vitamin B12 to grow. So if you see a place in the media after incubation with the growth, the more growth, that means some organism in that area is producing vitamin B12 and secreting out for the other microorganism. So in that way, we can isolate the microorganism which is producing metabolites. So in this test, we have, I will go through it one more time. We are taking the sample, then get diluted to get isolated colonies. Then it is plated in a minimal media without the particular metabolites. Then incubated with a orthotrophic microorganism. After incubation, there will be a zone of growth. If the any of the microorganisms in the sample is producing the particular metabolites, the test organism will grow around that microorganism. In that way, we can isolate the organism producing particular metabolites. It can be used for the isolation of vitamins and amino acids producing microorganism. Finally, we have a method to isolate organic acid producers. Organic acids are most commonly used in the food industry. So methods are more or less same with some modification. We are taking a sample and then diluted to get isolated colonies. Then it is spread on agar plate. In this case, the peculiarity of agar plate is like it has a pH indicating dye, like neutral red. It will change the pH, the color based on the pH. Then incubated. After the incubation, if the any of the microorganism in the sample is producing an acid, what happened? The pH, the agar is already having a pH indicating dye. The color of the medium will change. So based on the color change, we can identify the microorganism which is producing an acid or alkali. In this way, we can isolate the microorganism from the nature which is able to produce organic acids. In this case, the sensitivity of the test can be adjusted by adjusting the buffering capacity of the medium. If the medium is having very less buffering capacity, that means it cannot resist the change in pH. So the test will become more sensitive. If the medium is having high buffering capacity, even if the microorganism producing a good amount of acids, it won't change the color of the medium because the medium is already having the buffering capacity. So in this case, the sensitivity of the test can be adjusted by adjusting the buffering capacity of the medium. So now we have three examples of direct selection. One is for the isolation of antibiotic producing microorganism, second is for the metabolite producing microorganism, and third is for organic acid producing microorganism. The most all these methods are similar with some modifications. Secondly, we have enrichment culture. It is mainly used for the isolation of for the indirect selection, but it is not always. We can use this method in a modified way to for the direct selection also. In all type of enrichment culture, you have already known the enrichment media. Uh, so in the enrichment culture, we are increasing the number of desired microorganisms. So either we are incorporating an inhibitor or growth promoting compound in the media so that the number of that particular type of organism will be increased. In industrial sense, enrichment culture use cheap medium at high temperature. We are using cheap media because if we are isolating a microorganism using cheap media in the enrichment culture, the organism will be able to grow in the cheap media when we are using it for industrially. Then another thing is that the cheap medium means it will be a byproduct of some other industry that will be heavily contaminated or it have a very high microbial load. So if we are isolating a microorganism which is able to survive and able to produce a particular product in a cheap and contaminated media, that will be very resistant to contamination. So always we use cheap medium for enrichment culture, not the laboratory media. We are using the industrial media, like the media formulated from the waste product of other industry. And also we are using high temperature. I'm sure all of you know why we are using high temperature in the enrichment media. The enrichment culture can be done in two different modes, batch and continuous mode. Batch culture is like repeated subculturing. In first day, we are incubating the samples in a conical flask. We are incubating for the period, maybe for overnight. After the incubation, we are taking a small sample from the conical flask and incubating it into a fresh media. And again, we are incubating the fresh media. After overnight incubation, we are taking a small sample from the conical flask and incubate into another fresh media. And we go on like that. It's like repeated subculturing. In this type of repeated subculturing, selection is purely based on specific growth rate. Those organisms growing faster will be get selected. So at the end of 30 subculturing, the organism with the maximum growth rate will be survive. Anything else will be, all other organisms will be washed out in different stages of subculturing. This method is an excellent method when we are looking for a single cell protein, because when we are isolating a microorganism to produce used as a single cell protein, the organism should have maximum growth rate. So in batch culture and batch enrichment culture, we are selecting an organism with the maximum growth rate, not the maximum yield or maximum productivity. The only criteria is maximum growth rate. So if you are looking for an organism for single cell protein, it's the best method. But for nutrient, for the products, other products like antibiotics or amino acids, it is not the best method. For that, we have continuous enrichment culture. Continuous enrichment culture is like a chemostat. We are doing the enrichment continuously. We are adding medium continuously and we are withdrawing the medium. Continuous enrichment culture, selection is based on limiting substrate. We can control the growth rate using the dilution rate. In the later chapters, we will learn the kinetics of continuous culture. So the selection is based on limiting substrate. We can control the growth rate of the microorganism by manipulating the dilution rate. One advantage of continuous enrichment culture is that if you want to use a microorganism for continuous process, it's better to enrich it in a continuous culture. If you want to use a microorganism in batch culture in an industry, it's better to isolate in a batch mode. Same thing. One another advantage is if a bioprocess is done by two different microorganisms, it's very difficult to isolate them in a batch culture. But in continuous culture, we can isolate multiple microorganisms together. So it can isolate microbial consortia, a group of microorganisms doing same process. One downside of continuous culture is washout. For every continuous culture, the growth rate is equal to dilution rate. If you if you growth rate is increased, you can adjust it with the dilution rate. But sometimes what happens is like the growth rate will be low and the dilution rate will be high. Low growth but dilution is high. In that case, what happens is like all the microorganisms from the continuous culture will disappear. During time it will disappear because the dilution rate is high and the growth rate is low. That's called washout. All organisms from the fermenter is washed out because of the high uh, dilution rate. So that's the one disadvantage of this continuous enrichment culture, which can be avoided by three different strategies. One is we first do a batch culture to increase the number of microorganisms, then go for a continuous process. Or we can use a turbidostat. We already discussed that turbidostat is a continuous culture with a turbidometer. There are other options, two-stage chemostat. In two-stage chemostat, we have two different chemostat. One chemo from the first chemostat, we take the medium, withdraw the medium, add into the second chemostat, and then again do the continuous culture. So there will be two different continuous culture will be running simultaneously, and the second chemostat is using first chemostat withdrawing medium as the inoculum. So in that way also we can avoid washout. We will discuss the more details of the continuous culture later. So that's all for primary screening. Once we get the microorganism which is producing a useful product, we can go for the secondary screening. In the secondary screening, we are comparing the microorganism, the strain, with the ideal characters of a strain. We already discussed the ideal characters of an industrial strain. Secondary screening basically done in three different like phases. First, we assess the microorganism, the strain, whether it is the properties of strain. Then we assess the properties of the product. Then we assess the real industrial value of the whole process. So first we look into the strain. First thing we want to do is the systematic classification. Once we found a microorganism which is producing an antibiotic, what we do, we try to identify the microorganism. In earlier days, we used the biochemical test and the microbiological test to identify the microorganism, but which take long time. Now we have the molecular biology techniques like rDNA sequencing. So we go for the systematic classification of microorganism to know that what is the genus, what is the species, what is the like variant of the microorganism. Once we know the systematic classification of microorganism, we will know like whether the microorganism is a new microorganism or a old one, it's a new variety or not. So we can know so much things about the microorganism once we've done the systematic classification. Then if the organism is unknown, we got a new organism. If the variant is unknown, we can publish it. After the classification, we study the characteristics of the microorganism, both the microbial characteristics and the biochemical characteristics. If the organism is already known, it's easy to characterize because already the characteristics are elucidated. So the microbial characteristics, we will culture the microorganisms in different culture medium. We study every parameters, the shape, size, the arrangements. Then we will go for a biochemical test. There's a trait utilization, whether it is using lactose, whether it is using starch. Everything can be studied in the characterization. After that, we'll go for growth and product kinetics, how it is growing, what are the phases of growth, how well it is growing, what are the inhibiting factors, how the product is formed, in which stage the product is formed. Everything will be studied. Then we have the genetic stability test, whether the organism is reproducible, the test and production and growth is reproducible. Then we compare it with the existing strain. If we have a new antibiotic producer, we compare it with the already or organism which already we already have which produce antibiotics, so that we will get a comparative analysis of the organism. Then we do the pathogenicity test, whether it is producing any disease to the humans, animals, environment, anything. Then we estimate the yield potential, what will be the maximum yield if you do it industrially. Then we have to optimize the physiochemical and nutritional conditions. What is the optimum pH, optimum temperature, what is the aeration and agitation needed for the organism, then the nutritional characteristics like what are the raw material it can be used, what is the precursivity needed for the product formation. So everything about the organism will be studied. Then we have the product characterization. We need to characterize the product both qualitatively and quantitatively. The novelty of the product should be measured, whether it is a new product or the already existing product or the transformational product. Maybe the newly isolated product will be very similar to another product already in the market. Then toxicity studies, whether it is toxic, what is the toxicity level, what is the LD50, everything need to be identified if it is toxic. And identification of intermediates and byproducts so that we can modify it to get better productivity. Transformation potential, whether the microorganism is transforming or altering the product in some other ways. If it is doing it, whether it is good or not, we need to identify. Chemical stability of the product, how much time it can be in the medium, what is the shelf life of the compound, what are the microorganisms and enzymes responsible for the degradation of the compound. Then physiochemical properties of the product. Once we get the physiochemical properties of the product, we can devise the method for the downstream processing of the compound. Once we have done it, we will study the industrial feasibility of the whole process. We assess the commercial value, what is the market potential and everything. We look into the possible genetic engineering options. Are we able to transfer the genes from this microorganism to another microorganism or this animal to another microorganism to get better productivity? What are the manipulations that can be done to improve productivity and yield? IPR associated, if the organism is already known or the product is already known, whether anybody else is having a patent on the compound. If somebody else is having the patent, we cannot do anything with that product without permission. Market survey, if we produce this product industrially, what is the potential market, how much profit we are going to get. Production economics will be studied, the EDP stuff. They will do everything before starting an industry. So that's all for the secondary screening. In the secondary screening, as we discussed, we will look into the microbial strain, we look into the product, and we will product into the industrial feasibility. Secondary screening, as you already understand, it's a very complicated process. We are comparing all the properties of the new microorganism and its product with the ideal characters. We cannot do it in a single step. We cannot do a single test and say that the secondary screening is over. It's a multi-step process and a multiple collaborative process. But some of the methods which can be used, which is used in the industry for secondary screening, the simple steps are there. One example is giant colony technique. You will do the laboratory practical of this giant colony technique. It is used for the assessment of antibiotic spectrum of an organism in secondary screening. I will just go through the step. Here you can see a line of microorganism. In the primary screening, if you get a microorganism which is producing an antibiotic, in the secondary screening, if you want to know what is the spectrum of antibiotic, whether it's a broad spectrum or it's a narrow spectrum, what all the microorganism is going to inhibited by this antibiotic. So what we do, we will take the antibiotic producing microorganism, and we will incubate like this, like a straight line, right from one end to the another end. Then we inoculate the test organism one by one from the edge of the bedded plate to this line. We first one will be incubated from one here to here, second organism, third organism, fourth organism, so on and so forth. We inoculate multiple organisms. This will be a gram, maybe it is a gram positive organism, it will be a gram negative, it may be a mycoplasma, different microorganisms. After incubation, we will get a plate like this. So what do you see? The first organism is totally incubated. We have incubator from this end to here, but you cannot see any line of growth. So it is totally incubated. This one is incubated to an extent. It is not that much incubated. This organism is not at all incubated. There is only a small difference in growth. So by using this technique, we can identify the spectrum of antibiotic incubation in a single test. It's a sample of secondary screening method. It is we cannot do this test and say that every secondary screening is over. It's just a sample. And in secondary screening, several molecular biology techniques are used in these days to improve the screening because we have a very high diversity of microorganisms. There are billions of microorganisms even from a gram of soil. It's very difficult to screen all of them. So many molecular biology techniques we are using right now for these screening methods. The molecular biology, they provide test organisms, it provides gene cloning options, it provides reporter gene assay, then detection of organism using molecular probes and immunoassay. So these all techniques helps screening in a better way. So this all for today. In the next section, we are going to discuss how we preserve this isolated microorganism. Thank you.